So first, I want to thank all of you for attending today on a nice spring day. I want to commend you for spending some time here learning about your disease or learning about this for a loved one. Patients who are invested in their healthcare have the best outcomes, so glad you're all here. All right, this is the standard program disclosure slide from the Crohn's and Colitis Foundation. These are my disclosures. We're going to talk today about new therapies and clinical trials, and I do conduct a lot of clinical trials. I also want to mention that a lot of the slides I'm using were made by one of my colleagues, Dr. Lord, so I want to give him credit. All right, so today, this is what we're going to talk about. We're going to discuss the new and emerging therapies with a focus on 3 classes of medications: the IL-23 antagonists, JAK inhibitors, and the S1P1 receptor modulators. And then we're going to understand what clinical trials are all about, the steps of drug development, how trials are designed, and what it might look like if you were to consider participating in a clinical trial. So you can look at this kind of timeline of medication introduction for inflammatory bowel disease. And you can see in the 50s and 60s, we pioneered our first therapies: steroids, immunomodulators, and aminosalicylates. And it was about 40 or 50 years before we introduced the next class of medications, the TNF antagonists. And then really just in the past 8 years, we've had 4 new drug classes that have been approved, and there are a lot in the pipeline. And so we're going to focus really on the last 3 here, and we're going to go over the data behind them and kind of their relative advantages and potential drawbacks. So starting with the IL-23 antagonists. These are biologic drugs, which are large proteins. They're antibodies. Antibodies or large proteins, if you ingest them orally, get broken down by digestive enzymes, and they won't be absorbed intact. So these have to be given as an IV or an injection. They're based on human antibodies, like all the other biologics that we use for inflammatory bowel disease. And these target a cytokine called interleukin 23. Cytokines are chemical signals that inflammatory cells use to communicate with one another. And this one seems to be particularly important in certain immune-mediated diseases, including Crohn's disease, ulcerative colitis, and psoriasis as well. So if you look at this diagram here, you can see that interleukin 12 and 23, see if does the math show up on there? Yeah. So interleukin 23 is made of 2 subunits, P19 and P40. Interleukin 12, also made of 2 subunits, and shares this P40. Ustekinumab is an antibody that targets P40 and actually blocks interleukin 12 and 23. Interestingly, when the drug was developed for inflammatory bowel disease, it was thought that it would be an effective therapy because of blockade of interleukin 12. Turns out that's not the case, and while it's a very effective drug, it's all due to the interleukin 23 activity. There are now drugs that are available on the market for other conditions and under study for inflammatory bowel disease that target P19 and selectively block interleukin 23, and these are the drugs listed here. At least these are the generic names. Guselkumab is on the market for psoriasis, Tremfya, and risankizumab is on the market also for psoriasis. It's called Skyrizi. So you can see here that there are 4 drugs that are under development, all in this interleukin 23 class. And these are the phase of development. And when we talk later in this talk about the phases of drug development, you'll learn that phase 3 is really the last step before a drug gets approved. So most of these drugs are already in phase 3 trials for both Crohn's and ulcerative colitis. This is the data that have been released, at least in conferences and in a press release, for risankizumab for Crohn's disease, and it's quite effective. You can see for induction, the 2 doses of study drug were much better than placebo, both in the advanced trial and the Motivate trial. So these are 2 separate studies. The Fortify trial is the maintenance study. So the patients who responded to the induction studies were then allowed to enroll in this maintenance study, and you can see the drug performed much better than those who got induction only without maintenance of remission. So I know it's hard to compare, but I can tell you these numbers are better than what we see for most drugs for Crohn's and ulcerative colitis. You'll have to take my word for it. We're also starting to look at potentially using these interleukin 23 antagonists in combination with some of our other medicines like TNF antagonists. This is a study called the Vega study that looked at golimumab, which is a TNF antagonist, guselkumab, an IL-23 blocker, or the combination of the two. And you can see for clinical remission, endoscopic normalization, and histologic remission, which means the biopsies don't show any inflammation, that the combination of the two was better. And in fact, we have a lot of studies going on now exploring different combinations of drug classes. An anti-integrin therapy with a JAK inhibitor, an anti-integrin therapy with a TNF antagonist, this which is IL-23 and a TNF antagonist. So we're learning more not only about new therapies, but how to potentially use them in combination with existing therapies to get them to work even better. All right, moving on to the next class of medications that's new and emerging, the janus kinase inhibitors, which are collectively called JAK-inhibs. These are small molecule medicines, so they're available in pill form. And these block the enzymatic activity of proteins used to deliver signals from the cytokine. So I have a diagram here on the next slide, I believe, where we can go over that. There are a bunch of different cytokines, these chemicals that white blood cells use to communicate with one another, but they all signal through 4 janus kinases. They're called JAK1, 2, and 3, and then TYK2, or tyrosine kinase 2. So if you block one of these, you can block multiple cytokines, all with one medication. So here's a diagram that illustrates this concept. The cytokine in green binds to the receptor on the cell surface. That then activates through this JAK-STAT pathway, phosphorylation of STAT proteins, signal transducers and activators of transcription that carry that signal into the nucleus of the cell, which then causes the cell to take action. So binding of the cytokine requires this process to be able to actually carry out any effect of that signal. And JAK inhibitors block that step. You can see here that these are the cytokines that signal through these various JAK combinations. So interleukin 7 signals through JAK1 and 3, and so on and so down. So if you were to block JAK1, you can block signaling of all of these interleukins. If you block TYK2 in green, you block these lower ones. So you can see that different JAK inhibitors may have different activity, different side effects, as well as different effectiveness. One of the good things about this class of medications is that they work really quickly. This is data with tofacitinib, which is approved for treatment of ulcerative colitis. And these are symptom scores for patients. And you can see that by day 3, there was already a statistically significant difference between the treatment group and placebo. That's pretty rapid. Some of the medicines we use take 3 to 6 months to really see a benefit. And this, we see benefit within 3 days. It does come with some downside, though. There is a black box warning for tofacitinib, and until we know otherwise that this is not a class effect, we're probably going to see the same warnings with the other JAK inhibitors. For serious infections, all-cause mortality, malignancies, particularly skin cancers and lymphomas, major adverse cardiovascular events, which includes heart attacks and strokes, and thrombosis. Now, I can tell you all of these events are rare, but we're seen more commonly with this drug than with a reference population that was taking a TNF antagonist. So there's advantages and disadvantages to these medications. And this is the status of the JAK inhibitors that are currently under study. Upadacitinib, I should mention, was just FDA approved 2 months ago now, a month and a half ago, for ulcerative colitis. The phase 3 trials are underway in Crohn's disease. The maintenance data has already been released and showed benefit. So within a year, we should see probably the maintenance data, and most likely that drug will be approved based on the data that I've seen. These other drugs are in phase 2 or phase 3. All right, moving on to the third class of medications in new and emerging therapies. S1P1 agonists. So these are also pills. They're small molecules, which makes them much easier to take. And what these drugs do is that they activate the S1P1 receptor, which is used by lymphocytes to sort of sniff their way out of the lymph node. So this is one of the slides given to me by Dr. Lord. So all the animation is his. Basically what happens is that your immune cells, lymphocytes, make their way into a lymph node where they get educated and learn what antigen to recognize, and then they have to find their way out of the lymph node and back into the blood vessels to circulate and find their target. And one of the ways they find their way out is by sort of sniffing a gradient here of S1P. It's sort of like a salmon that finds its way upstream by sniffing the current. So what does this drug do? Well, this drug basically floods the lymph node with something similar to S1P so that the lymphocytes can't find the gradient. They don't know which way to go, and they get trapped or stuck in the lymph node. What that means is that those lymphocytes are not circulating, and we see a reduction in circulating lymphocyte numbers. Lymphocytes are used to fight infections, so there's concern for infection, although I can tell you in the study of ozanimod for ulcerative colitis, we didn't really actually see that many infections, but we don't have a lot of long-term safety data, so that's one of the unanswered questions. So ozanimod is already approved, the first drug in this class for ulcerative colitis. It's also effective for multiple sclerosis and approved for that as well. It was effective for clinical remission, clinical response, endoscopic improvement, and mucosal healing, which is complete endoscopic remission, both in induction and maintenance. What are the potential concerns? As we already talked about, the drop in lymphocytes may translate into an increased risk of infections. There were some patients who had heart rhythm disorders, particularly slow heart rate rhythm disorders. So that requires some monitoring, and for patients with pre-existing cardiac disease, might not be the best treatment option. Patients in this study were immune to shingles before enrollment, but there's likely an increased risk of shingles, and we recommend vaccination for that in susceptible patients. And there were some other adverse events that were seen more commonly with this drug that are not common, but are important and involve some discussion if we're going to prescribe this, and that we're going to be on the lookout to see if these things are also seen with other similar drugs in the class. So this is the current state of S1P1 agonists. Ozanimod has been already approved for ulcerative colitis. Phase 3 trials are ongoing in Crohn's disease. Etrasimod is a little bit further behind, but in phase 2 and phase 3. All right, so to summarize the new and emerging therapies, the interleukin 23 blockers are available as injections or infusions, pretty far along in study. They're safe, they're effective, and they're similar to a drug that's already on the market, ustekinumab or Stelara, although slightly more targeted. And I can tell you, in the case of psoriasis, these IL-23 blockers were compared head-to-head against Stelara and were more effective. Studies of head-to-head these drugs versus Stelara and Crohn's disease are currently underway. In terms of the JAK-inhibs, these are very fast-acting medicines. They're very effective. I've been using that upadacitinib in clinical trials now for 7 or 8 years, and I have some patients who are still on the medication. There are some potential safety issues, at least that we've seen with tofacitinib, a similar drug that we need to explore further to see if it's a class effect. And then the S1P1 agonists, these are also pills that are available now for ulcerative colitis. And I think we just need a little bit more long-term safety data, but hopefully these will pan out for Crohn's disease as well. All right, moving on, we're going to switch gears now and talk about clinical trials. So clinical trials are studies where we test these drugs to see if they work, and we monitor for any side effects. All of the therapies that are available had to go through clinical trials. This is really the foundation of our understanding of whether or not the drugs work, and any better than placebo or better than some comparator, and whether or not they're safe, whether or not the side effects we see occur more commonly than with placebo. So what are the steps of drug development? How do we decide, which drugs to test for inflammatory bowel disease? Well, it starts with preclinical studies. So this is basic science research. Investigators in a research lab study the immune system. They use models of inflammatory bowel disease, or they may get, cells or tissue biopsies or blood specimens from patients in this room. And they study what's different about the immune system in patients with inflammatory bowel disease versus people with a normal functioning gut immune system. And they try to identify potential targets that might be something we can intervene on to change the disease. If investigators identify something that they think is a promising drug, then they have to apply to the FDA. And it undergoes a safety review of all the, the chemistry and pharmacokinetics of the drug. And the FDA has to approve the study of this drug in people. We then go on to phase 1 studies. Phase 1 studies really are looking for safety or toxicity rather than looking for effectiveness of the drug. So usually we recruit a small group of people. They're considered healthy volunteers. And they may or may not actually have IBD. They may just be totally healthy people. We give them the drug and we measure where the drug goes in the body, where does it distribute, what's the half-life, is it eliminated by the kidneys, by the liver, are there any obvious signs of toxicity? These trials are not designed for effectiveness. They're really just sort of giving a test to make sure that there's no obvious safety signals. We then move on to phase 2, where the main question that researchers are asking in phase 2 is there a signal of effectiveness? Typically, these are dose-ranging studies that take the patients with the disease in question, in this case, inflammatory bowel disease, typically anywhere from 50 to maybe 200 people, and generally study multiple doses with some placebo group to compare to, so to know if the improvements you see are any better than what we would see with placebo. After phase 2, if the drug looks like it has a signal of effectiveness, then after the multiple doses, you can select what you think is the most effective dose and safe dose. Then you go on to phase 3, where you're really looking to see, is this drug better than placebo or better than standard of care? For these studies, we recruit several hundred patients, and the goal really is to demonstrate the effectiveness and safety of the medication. These are the foundational trials, the pivotal trials that drug approval hinges on. If the drugs pass muster in phase 3, then they get approval. And for most trials in inflammatory bowel disease, they're designed with 2 phases. There's an induction phase where we take patients with active disease, generally moderately to severely active disease, and see, can we get them into remission? And then we study those patients who have responded and randomize them to continuing or stopping the medication to see if you need to continue the medicine to continue to see benefit. FDA approval is contingent on completion of 2 phase 3 studies that show both effectiveness and safety. And then after the drug is approved, oftentimes drug companies will continue to study it to answer questions about long-term safety, maybe rare side effects. They continue to monitor the drug and in real-life treatment scenarios. So how are clinical trials designed? They have a very detailed protocol, oftentimes a couple hundred pages, that are painful to read through, I'll tell you. And it describes everything about the study drug, the rationale for using it, what's known about the safety of it. It describes the patients that are eligible to participate in this trial. So what is the patient population and the disease that's being studied? A timeline of all the study treatments, as well as the study assessments. How are we going to check and see whether or not the drug is working? And the treatment algorithm and the statistical analysis plan are also included in here. What percentage of patients get placebo? What happens to them if they don't respond? All of this is detailed in the protocol. There are some common elements that are in protocols, and I've already been using some of this vocabulary, so it's probably good for me to review some of the definitions. A control group is a group of patients in the study that do not receive the intervention that you're looking at. And you use that to compare. Whenever you do a new intervention, you want to be able to compare it to see is it better or worse than either standard care or better or worse than than placebo. Placebo is an inactive compound that is meant to look identical to the study drug, and it's given to some patients in the group so that we know, is the study drug actually better than doing nothing at all? And if we have side effects that we see, are those side effects actually more common with the drug than with placebo? Randomization is a process where the treatment groups are randomly assigned. So that way we can make sure that all the groups are identical with respect to any factors that might predict the outcome. If the investigators had to choose, who gets the drug and who gets placebo, we might choose, I don't know, all of our sicker patients, we might choose to get the study drug, and our less sick patients get placebo, and then you do comparison, but you're comparing apples to oranges. So randomization makes sure that the groups are even, just because it's a computer algorithm that spits out a treatment assignment. There's no human intervention there. And blinding is a technique that is used to reduce bias. So sometimes patients are blinded in the study. You may not know which treatment group you've been assigned to. You may not know if the IV infusion you're getting is just saline or if it has an active drug in it, or the same with the pill, if it's just a sugar pill or if it has active drug. Sometimes the investigators are blinded too, in which case we call it double blinding, so that we're not biased when we make an assessment about whether or not the drug's working or whether or not you're having a side effect that might be attributable to the drug. Certain studies you can't do blinding. Surgical studies, for example, it's oftentimes hard to blind the surgeon. This is an example schematic of a trial protocol. This is not a real trial, I just made this up. But there's a screening phase. After screening, in this trial, randomization, I said, was 3-2. So 3 patients get study drug, 2 in placebo. And it's important, if you're considering a clinical trial, to think about what are your odds of getting placebo. At 12 weeks, patients get a colonoscopy, and then they get re-randomized 1-to-1. So those who got drug have a 50% chance. This is supposed to be a different dose of the drug. I think there are some formatting errors from moving the presentation over to this computer. And then patients, after completing 52 weeks, have 12 weeks of a safety follow-up. This is a somewhat typical design for many studies. So if you do want to participate in a clinical trial, the first step is really informed consent. This is a very important part of the process. This is where you're given written information about the study, about why the study is being done, what the intervention is, the risks, the benefits, your rights and responsibilities as a patient. You take this home, you read it, you mull it over, you take as much time as you want, you discuss with whoever you might want, friends, family, primary care provider, whatever you want to do. When you are ready to proceed, then you go over this document with one of the study coordinators, and they walk you through it to make sure you've read it, to make sure you understand everything. This is your chance to ask questions and see if this is what you want to do. Just some things to think about if you're considering participating. What are the risks and benefits to you as an individual? Not just generically, but to you, given your health conditions. Is there a placebo, and what are the odds of receiving placebo? How long does the trial last? These are a significant time commitment. Are you going to be able to do it? Are you planning to move away in the next 6 months? What's the timeline of treatments and assessments? Am I going to need a colonoscopy? How many colonoscopies am I going to need over the course of the trial? What are the potential financial costs of the study? And what other treatment options are available? If I don't do this, what are my other treatment options? If you do decide to sign the informed consent, and then you undergo a phase that's called screening. To be eligible for the trial, you have to meet all the eligibility criteria, and there's quite a long list of them. Study team reviews your history to see if you have any pre-existing health conditions that might exclude you from participating. You undergo a bunch of blood tests and maybe some other tests, chest x-rays, EKGs. This is to screen and identify if you maybe have any health conditions that exist before the study. Maybe they don't exclude you from participating, but it's good to know if they're there, because then if something happens during the trial and we find you have a health condition, it's good to know that it was present at the beginning. Some of those health conditions that we find might exclude you from participating. Most trials for inflammatory bowel disease are going to include a colonoscopy as part of the screening. And the colonoscopy has to show moderate to severe active disease. And typically that's a video recorded and sent to a blinded investigator who reviews it, so that the doctor doing the colonoscopy isn't tempted to round up or down to get you in or out of a trial. It's sent to someone with no identifying information who reads these studies as part of a company that offers this service. So these are some examples of eligibility criteria. There's usually a list of about 50 inclusion and exclusion criteria, but these are typical ones. So for a study of Crohn's, you would need to have a diagnosis of Crohn's typically for at least 3 months. They may want you to have tried at least one other therapy before you enroll in this trial. In this trial, it's for age 18 and older and capable of providing informed consent, and you have to have moderate to severe disease, both based on symptoms and colonoscopy findings. Examples of exclusion criteria: most studies will exclude pregnant patients for safety reasons. Maybe if you have an active infection, that might exclude you. Prior surgery to remove most or all of the colon might exclude you. Active or recent diagnosis of cancer, and maybe certain pre-existing health conditions that might make participation unsafe. These are just some examples. If you make it through screening, so you meet all the inclusion criteria and exclusion criteria, then you participate in the trial. So you're going to start to receive the study intervention, and you're going to start to undergo all of those assessments of effectiveness and safety. You'll likely be randomized and blinded and assigned your treatment group. The protocol is what dictates how frequently you're seen, at what time points, and what happens at each visit. But you can expect to be seen pretty frequently. These trials are really designed for safety. So we see patients pretty regularly, maybe right after your first dose at 2 weeks, 4 weeks, 6 weeks. We might see you, every couple weeks. And then probably every 4 weeks out to a year is very common. And you can expect that the visits take a little bit longer than your standard visit with your provider. And we're going to ask questions head to toe. Is your scalp itch, any visual changes, all the way down to ingrown toenails, to see if there may be any potential side effects. Between visits, for most of these studies, you're going to get some sort of an electronic diary. So you're going to go home and record your symptoms. It may ask about your abdominal pain, your number of bowel movements, how many of them are loose, bloody, your general well-being. And all that data is going to then be captured by the investigators and used for analysis. That's how we knew, for example, that tofacitinib started to improve symptoms within 3 days, was based on those diaries. And then at some point the study ends. You're not in the study for life. Every study has a defined endpoint. Once you've gone through all the assessments and time points, if it's a 52-week study, after week 52, you're done with the study. Some studies do have long-term extensions. So, and this is something to think about as part of the informed consent. If the drug works for you at the end of a year, what's going to happen? Will you still have access to it? Many times you can roll over into what's called a long-term extension trial, where then you're receiving the study drug on an ongoing basis. But that's technically a separate study. It involves another informed consent, and if you choose to, you can participate in that if it is offered. But it's good to know that up front when you're thinking about participating. You can choose to withdraw from a study at any time for any reason without any punishment or prejudice. Your doctor can't withhold other treatments from you or somehow be mad at you or whatever. You can withdraw at any time for any reason. The study sponsor and the investigator can also withdraw you from the study. So they may have learned information about the study drug that they think makes it unsafe for you to participate. Maybe there's a side effect and it's in people just like you, or you have a health condition that may be worsened by this. So either the investigator or the sponsor, the company paying for the trial, can also withdraw patients from the study. You should know that safety is baked into this process from the get-go. Before starting the trial, the protocol has to be reviewed by the FDA. And each site that is enrolling patients, their local ethics board has to review the protocol and make sure that this is something ethical to do. And those bodies get periodic reports about the conduct of the trial and the safety. In terms of site selection, each site that's going to conduct the study has to be visited and reviewed and the facility toured to make sure we've got the resources, the equipment, all the staff have to have the knowledge, training, expertise. We have to submit everyone's CVs. They have to undergo training for the specific protocols and learn about the drug. This is before we can enroll a single patient. Once we start recruiting, your local investigator who's seeing you at all your visits, as well as the coordinators and the local ethics board, the institutional review board, are going to monitor all the data from all of your visits, and they're there to ensure your safety. The clinical research organization that conducts the study has monitors that come out and they go through all of our records with a fine-tooth comb, and they really find every I that's not dotted and every T that's not crossed. And then the sponsor reviews all of this data as well. And there's an independent data safety monitoring board that reviews the data. So trust me to say that safety is not really much of a concern with these studies. There are multiple redundant layers of oversight. And the whole process, while it's bureaucratic and requires a lot of paperwork, it really does protect patient safety, which is our #1 priority. So what are the typical advantages and disadvantages to participating? I would say the advantages are early access to some promising treatments. It may be that you've already tried everything that's already approved for your condition. You could think about enrolling in one of the trials of these therapies that I talked about that are now in phase 2 and phase 3. And in most of the trials, all of the study-related treatments and the assessments are all covered by the study. So you don't have to worry about insurance copays and all that stuff. Every study visit, every colonoscopy is paid for by the study. The main disadvantages, it's a significant time commitment. And you need to be prepared for that. You need to have childcare arrangements or time off work arrangements to be able to participate. There's a chance you might get placebo. You have to think about whether or not that's something that is acceptable for you and how bad your symptoms are if you're potentially not on therapy for a little while. We are studying these drugs because the effectiveness and the safety hasn't been fully established. So while many of them are really promising, there is a reason why we do have to study them, and some drugs that look good in phase 2 do fall flat in phase 3. So there's, you need to be aware that the safety and the effectiveness are still not fully established. And there are family planning implications. So certainly for females of childbearing potential, in most studies, we ask that you do not become pregnant during the study and be on some form of birth control. If you do become pregnant, you're typically automatically disenrolled from the study. Some studies have similar restrictions for male partners, that they not try to conceive a child and use barrier methods of protection. So how can you identify trials that are right for you? Well, start by talking to your healthcare provider. That's where I would always go about treatment decisions. And in general, if you're doing well on your therapy, stick with it. Don't be looking for something else that sounds better if you're doing well. But if you're not doing well, then talk to your provider about the options. Maybe you can change the dose of the drug you're on. Maybe you can add something to the drug you're on. Maybe you can switch to another FDA-approved therapy. Maybe a clinical trial is right for you, but start by talking to your healthcare provider. And in some situations, enrollment in a clinical trial may be the right option. These are some resources, and I think you'll probably have online access to the slides. So if you want to look up trials that are available or more about what clinical trial participation is like, you can follow these links. That's it. Thanks.